So like Harold said, my name is Stephanie Karhoff. I started last April as the Extension educator in Williams County. So the question I want to pose today is whether this Fallow Syndrome, will it be a problem in 2020? So when we have fields left unplanted or fallow, we can see a decline in beneficial mycorrhizal fungi. And that phenomenon is commonly described as fallow soil syndrome or fallow syndrome. And it doesn't need said that it's a concern going into the 2020 growing season because wet weather conditions prevented Ohio farmers from planting over 1.5 million acres this past year. So we have a lot of acres that were left idle that could be susceptible to this issue. The main symptoms that are typically associated with fallow syndrome are stunting of the whole plant as well as phosphorus deficiency. So you can see in this picture we have that classic purpling for our phosphorus deficiency. And the reason for this is that mycorrhizal fungi is a key driver in plant fertility. So it's increasing or extending our root system through structures called hyphae. So these are thread-like structures that are going to extend that root system by up to 40 times in some cases. And that's helping the host plant scavenge for nutrients. So in these photos here, it's from a publication. On the left side is a cherry plum tree. On the right is a leek plant. So those darker lines, that's the plant root. And then the thinner lines, that's the hyphae, those thread-like structures that are produced by the fungus. So you can see that the hyphae are really helping those roots go further and hopefully scavenge nutrients more successfully. However, mycorrhizal fungi require a living host to grow and reproduce. So the start of the life cycle for these fungi is germination of a spore. So you can think of it just like our seeds in the ground. That spore is going to germinate and then receive signals from those plant hosts. And once it's received those signals, develop those hyphae, those thread-like structures. And it's this part of the life cycle that requires a living host. So once it attaches to the root surface with these structures called hyphopodiums, it's going to attach to the root surface, penetrate the root cells, and at this point, it can start developing more and more hyphae until we get to the point where these tree-like structures called arbuscles can form. And these are going to serve as the point of nutrient transfer between the plant and the fungus. So in this symbiotic relationship, the fungus is going to receive carbon from the plant and in exchange is going to be providing nutrients to the host. And then once that's complete, it can go ahead and start in the beginning where it's developing more spores. So will fallow syndrome be a problem in 2020? I'm going to use the classic cop-out and say, well, it depends. And let's go into some of those factors that could be at play. So when Harold asked me to present on this, it stemmed from just a basic blog post I had done in Williams County. And I was a little bit hesitant to agree just because it's not a common problem in our region and not one that I've experienced firsthand in my career thus far. So when starting these slides, I had more questions than answers, to be honest. So this is the whiteboard in my office when I started writing those questions down on what variables could either increase or decrease our likelihood of fallow soil syndrome being a problem in 2020. And so some of these I'll touch on some past research that I've found, but they include what kind of tillage do we have, type, duration, frequency of that tillage, do we have residue in our fields that are present, what kind of weather conditions do we have in the winter during that fallow period, did the soil flood, what's our level of phosphorus in the soil, and did we get a cover crop in? So I relied on the fact that history typically repeats itself. So when I was looking for answers, I went to some on-farm research that was done by University of Nebraska that was brought upon after the Great Flood of 1993. So this flood occurred from May to September of 1993. The Mississippi and Missouri rivers and their tributaries were the main water areas affected, and it touched and affected North Dakota, South Dakota, Nebraska, Kansas, Minnesota, Iowa, Wisconsin, and Illinois. So these images that we're looking at here are satellite. So on the left here is the Mississippi River near the St. Louis area in August 1991, so normal conditions for August. And then on the right here, we have that same area, but in August of 1993. So you can really see how severe that flooding was. So the on-farm study that they did looked at whether or not mycorrhizal colonization decreased after that flooding and fallow period in 93. So what they did was they took soil and plant samples from 3 different sites. 2 of those in Iowa and 1 in Missouri. And at each one of those sites, there was 1 field that was flooded in 1993 and left fallow, so prevented plant situation, and an adjacent field that was not flooded and was able to be planted as normal. That kind of served as a control in this case. So they measured how much of the roots were colonized by these beneficial fungi, as well as phosphorus uptake and final yield. And it wasn't replicated, but each site was given a different treatment just based off of the farmers' practices for starter phosphorus. And that ranged from 25 to 80 pounds per acre. But I'll emphasize that each site had a different rate and it wasn't replicated within site, so it's not something we can get a lot of answers from. So I just adapted these graphs from that data. So here on the x-axis, we have their different sampling dates. So April 26th, 27th, May 25th, 26th, so after planting in 1994, and then July 15th, 16th was their final sampling date for soil and plant samples until they took final yield. On the y-axis is percent of those roots that were collected to what of those roots were colonized by that beneficial fungi. So looking at that first sampling date, flooded fields had 68% less colonization at that beginning of the growing season compared to the fields that were not flooded. Looking forward after planting, so now both fields have a living host for those fungi potentially. We still saw a 65% less colonization in May, comparing those flooded to non-flooded fields. This is where I think the story gets a little bit more interesting. So there's still a big gap between our flooded and our not flooded fields, 48%. But what I saw, what I thought was optimistic was you can see that slope is increasing for the gray or not flooded fields, showing that those mycorrhizal fungi are rebounding somewhat. And then final yield, there was a 12 bushel per acre difference between the flooded and not flooded fields. So even though we're seeing that rebound, there was a yield hit for those flooded fields.
Was that significant? Was that a significant difference in yield?
So the question was that a significant difference in yield? Across sites, yes. I will say, so these points are those 3 sites averaged, and there was 1 site where it had 2 pairs of yield. So in this paper, they did a t-test looking individually between those sites. So just comparing the flooded field to not flooded field. So in those comparisons, all of them were significant, but they were looking at within a location. So did mycorrhizal colonization decrease due to flooding in 1993 in 94? What they found was yes. So that lack of host plant was a detriment and did cause a decline. But from April to July, there was a 62% increase in that fungi in those flooding fields, suggesting that those fungi do have the ability to be rebuilt and rebound. So I mentioned that they had different starter phosphate rates at each location. It varied across location, but most did show some sort of early phosphorus deficiency, and that fits with the earlier slide where we had those hyphal networks extending our root system and our ability to uptake phosphorus. But even those that had the higher rates of 60 to 80 pounds per acre still saw a 7 to 16 bushel per acre difference. And what was common across all sites, that broadcast P was ineffective. So if you're going to do the starter P, make sure you're banding. So we know that a host plant is going to cause a decline in our number of beneficial fungi, but what other management decisions that we made in 2019 could affect us moving into 2020? So two of those that we'll look at next. One is soil disturbance. So that'll be analogous to maybe some tillage work that's being done. And then also how much residue is available in your soils, in your fields for that fungi to attach to. So for this one, this was done in a field of corn in Canada. So they looked at how well that fungi can overwinter, so surviving through the winter months, whether it's attached to corn roots or whether it's detached from corn roots, and whether or not the soil was disturbed or not. So they did this just kind of man-made soil disturbance, but you could think of it as similar to maybe some fall tillage work. So they buried soil-filled pouches in that corn field, and those pouches either had coarse mesh or fine mesh. So that coarse is going to allow the roots to come in, so that has the presence of roots, whereas that fine mesh is going to exclude the roots. So you can think of it as no roots versus roots. And then they had 3 different treatments for disturbance. So the first one is they did not touch the pouches at all, so no disturbance. The second one, they took the pouches out and just disturbed the soil outside of the pouch. So kind of like a medium in the two. And then the third was the soil was disturbed both inside and outside of that pouch. So the most extreme disturbance in this case. So overall, there was more active hyphae in the presence of roots. So here on the y-axis is what they were measuring was they took all those hyphae from their soil samples and did a ratio of how many of those can actually actively infect plants and help us with their uptake. And then the x-axis is our fall values, so when they first put those pouches into the cornfield. And then in the spring, so remember after harvest is when they did those 3 different disturbance treatments. So these red arrows are pointing to those pouches that had the coarse mesh, so they did have roots in those pouches. So overall, the white bars are giving us more active hyphae than our gray bars, which is that fine mesh that's excluding the roots. So overall, there is more active hyphae in the presence of roots. And just looking at the overwinter survival of these fungi in a normal situation, so say you did get your corn, soybean out in 2019, but you did not have a cover crop going into those winter months prior to planting in 2020. Even with that, without disturbance, that active mycorrhizae is going to decrease by 33%, which I think is important to remember is we'll have the sharp decline because we don't have a living host, but in a normal year, we're still going to have some sort of drop just because of the overwintering months.
If you had a cover crop host, you could take care of that drop because you have an active growing host instead of a dead host.
Absolutely. So one of the slides I'll have later on when we're talking about managing it in the future, the best way you can is a fall cover crop. So having a living host, that way it can still be completing that life cycle. But if we don't have a living host, having that residue is still beneficial. So now comparing no disturbance to that medium treatment, so just some disturbance. Active hyphae decreased by 49%, moving from the fall to the spring. And then our most extreme disturbance, that's going to reduce it by 77%. So overall, disturbance affected it the most, but residue did come into play, and one of their hypotheses was that that residue is still providing some sort of energy supply to that fungi in the soil and letting it overwinter through the months. And then the main hypothesis for why that disturbance is causing a decrease in active hyphae is just because the Disturbance itself is breaking up those hyphal networks. So remember, all those thread-like structures, they're just getting broken into smaller and smaller pieces to the point where they're no longer functional. So you beat me to the punch, but looking at managing fallow soil syndrome, if it is a problem that we're seeing, hindsight's 2020, but the best thing you could have done if you had prevent plant acres was to get a cover crop in this fall. And keeping in mind, not all cover crops are equal in this case, so our brassicas are not going to serve as a host to mycorrhizal fungi. So keep that in mind. If you're using something like radishes, you might need to mix it in with other species, like oats, that could serve as a host. Let's say you did not get a cover crop planted and you did have prevent plant acres and you are concerned about fallow syndrome causing some P deficiency and yield hit in those fields. You can try planting crops that are less dependent on mycorrhizal fungi for nutrient uptake. So if we're comparing corn and soybean, soybean is less dependent on that mycorrhizae to uptake nutrients. And in general, shorter root systems are going to be more dependent on mycorrhizae to pick up nutrients. So just some additional thoughts. My opinion is that from the data in the 90s, these mycorrhizae can rebound, and the effects are likely limited to a single growing season. So I think 2020, be vigilant, scout for P deficiency early on in the season, but it's likely to be limited to 2020, assuming that we have a normal planting season this year. Maybe knock on wood if you got it. Lastly, no field or soil is equal. So you may have more resiliency in your soils compared to your neighbors, and that's going to come into play too. So the big unknown factor for a lot of these decisions is you probably don't know what your starting population is for these mycorrhizal fungi. So you may be starting at a higher level than, say, another field. So even with that 30-plus drop, you may be in still maintenance range, so to speak. Benefit of starter P is unclear from what I've seen, but if you are considering it, if you are worried, make sure you have your soil levels already known. So especially here in the western Lake Erie Basin, we don't want to be applying phosphorus if we're already above those limits, because at that point, it'll be in the soil, but it still won't be accessible to those plants. And I also challenge those that are worried about this occurring in their fields. It'd be a great opportunity to do similar research that Nebraska did in '94, looking at different starter P rates, if you have a field that was flooded or not flooded, or if you had one that you got a cover crop in and one that you didn't. Maybe we can get some plant and soil samples to get more data on this question and get a little bit more solidified on what our recommendations are. So with that, I really thank you for your attention. I'll welcome any questions or if you've experienced this in the past yourself.
On the studies that you used to help provide some background for you, did they, where the water levels dropped, go in with a later season cover crop to see what difference that would make for their next year going into hopefully a better crop season?
So the sites in 93, 94 in Iowa, they did not have any cover cropping, and it was conventional till. Any other questions?
So here in Ohio, you've had now perhaps more than a year without any living host, isn't it? Some fields for red flag. So could that not have a bigger impact than what you showed in the slide that seems that was only from the previous year to the next, or not?
So in that case, it'd be a similar situation. So there had flooding in 93 that prevented planting. So some of those fields weren't planted in 93, so similar to 2019 here. And those fields didn't have any living crop in them until spring of 94, so analogous to spring of 2020 for us. But those were only 3 sites in different locations, so that's why I challenge if anyone's worried about this, let's partner with Extension and see if we can get just some rough samples and maybe get similar numbers, or maybe we'll find something different.
Did you get any producers willing to plant something in the fall to see what difference it makes and run your own comparisons?
So we had some question was, were we able to get some growers to plant cover crops to compare differences in most likely mycorrhizal colonization between the two? We tried. There was a bit of a scramble, honestly, this year. I think a lot of us were playing catch up on getting things out and in time for farmers to respond to. So nothing on the cover cropping. So we could still look at starter P, or if you had adjacent fields that had a cover crop and no cover crop, that might still fit. But nothing in a strip trial fashion.